we're going to do this kinetic enzyme study. For the experiment, we're going to use amylase enzyme. And we're going to study the kinetic of the enzyme. We're going to construct a standard curve, and how standard curve will be used to determine the concentrations and the activity of the enzyme. So what we have at the front with me, I have the enzyme itself, which is the amylase enzyme. I have our substrate, which is the starch, and the concentration is at 0.2% starch. And then I have iodine as our dye, and I have here phosphate buffer at pH 7. With me, I have this test tube, all labeled for our first experiment. There are two experiments I'm going to do today. The first one is to prepare the standard curve. And then the second experiment is on the kinetic of amylase. So this particular experiment is application from all the previous experiments that you guys have learned in the previous lab sessions. This only the application for you guys have been taught. So another mechanism, another tools that are going to be involved, which will not be shown in this lab session, is the UV spectrophotometer. But then again, however, we will give you guys a mock sample, a mock result, which you guys need to plot, and those results is based on the UV spectrophotometer reading. In this particular experiment that I'm going to show is how important we have to use the standard curve to determine the enzyme activity. So for the first experiment, I'm going to construct the standard curve. All I need is the starch solution at various concentrations, and then I need water, I'm going to add 3.5 ml of water, and for this one I don't add any starch. The second one, I need 3.3 ml of water. The third tube, I need 3.1. The fourth tube, I need 2.9 ml. Fifth tube, I need 2.7. The sixth tube, I need 2.5 ml of water. And then for our sample, I need 2 ml of water. since I don't have to add any water, any starch solution for the first tube, so the second tube I need to add 200 microliters of starch. For the second tube, I need to add 400 ml of starch. In this instance, I'm not going to change the scale. I'm just going to add twice of 200 microliters of starch. For the fourth, I need 600 microliters. In this instance, I'm going to add 3 times of 200 microliters. I'm going to add 4 times 800 microliters of starch. And the last one, I'm going to add 1 ml of my starch. And for the sample, I'm going to add 1 ml as well. But remember, for our sample, I only have 2 ml of water. Now I'm going to add 1 ml of starch for our sample. And then I'm going to add 1 ml of phosphate buffer into each tube. And now I'm going to add acidic iodine into the tube. How much? By 500 microliter. 500 microliter. So if you look through, the one that doesn't have any starch solutions, the color is brighter than those who have the starch solutions. Differences in the concentration as well, you guys would also notice that the concentration between the two is going to be slightly different as well. If I compare the one that have 0.2 ml of starch with the one that have 1 ml of starch, there is a major difference in terms of color. So the one that have more starch, the color is brighter, the color is darker compared to the one that have 0.2 ml of starch. Clearly, this is much brighter compared to this one, since this one has less starch compared to this. So now, after this, we're going to measure it using spectrophotometer. But then again, I will give you guys the result of my reading for you guys to plot for the standard curve. And now we're going to determine our enzyme activity. So now we have our starch tubes with dye in it, and we already measured with the spectrophotometer for the standard curve plotting. And now we want to compare. We will use our sample, and then we're going to add the enzyme into our sample and observe the changes in terms of colors and also in terms of the absorbance using UV spectrophotometer. So what I need to do now, since I already prepared 1 ml of 0.2 starch solution and added 2 ml of distilled water, and I also have added 1 ml of phosphate buffer. To observe kinetic activity of our amylase enzyme, I'm going to prepare a sample using the starch solutions as well. So what I did here is I'm going to use 1 ml of starch solution the same as during the first experiment. Two ml of distilled water, and then 1 ml again with phosphate buffer. Instead of adding iodine now, I would add amylase first before adding iodine later on. So I would add 500 microliters of amylase solution. Which is our enzyme. And I will incubate for between 10 to 15 minutes at 37-degree water bath. Now I'm going to add 0.5 ml of iodine to stop the reaction. Now what you've seen here, the colors of iodine become slightly brighter, and if we compare with our standard curve, our standard of starch, we could put side by side which concentration are closer to our sample. This one doesn't have any starch. Of course, this one have a reduced starch that has been catalyzed by amylase. This one have 0.2 starch, and you guys notice this is slightly darker compared to this one, our sample. And of course, when we have even higher concentration of starch, it's become a lot darker. It is clearly that our sample reside between 0 starch and 0.2 starch. In the beginning, during the first video, I've shown you our 7 sample, which is even darker than the original. Even darker. So now, after adding the enzyme, the amount of starch has been reduced. Therefore, the color of iodine become a lot brighter compared to the previous one. So now I'm going to measure on the UV spectrophotometer and to get the absorbance values, and then I'm going to plot on the curve. For your task, I will give you the absorbance values, and you guys also need to plot the curve by yourself in a group. And we're going to compare, and please find what are the exact concentration of starch after went through catalytic process. And make sure to answer all the questions given in the lab manual. That's it for now. For the remaining will be continued by the other lecturer. Thank you.